Good morning, everybody. It is the last week of our term for Biology 120 Lab. Let's see what we've got going on. Last week you learned about carrying capacities of ecosystems and how many individuals could be supported by an ecosystem. And you learned about studying cohorts in a population, for example, how many children you have in a population versus how many elderly people you have in a population. So you're studying populations and how they interact with the ecosystems. This week's topic is how you measure populations. There are all different ways to do this, and if you think about an organization like the Department of Natural Resources or whatever you call it in your area, they have to have lots of ways to measure populations. So if you're measuring the population size of bald eagles, that's going to be completely different from feral hogs, deer, rats in a big city, human beings around the world. Yeah, so there are lots of different ways to measure population size. We're going to take a look at 3 this week. Transect, so you draw a line across an area and you measure out every so often and you see how many individuals you have of a particular species over that distance at those locations. So that's a transect. Quadrat is more of a roped off area. And so you look at the whole area to find out how many individuals you have. You might want to do this with fire ants or something like that. I don't know if you have fire ants in your area, but yeah, we've got them everywhere here in the South. Mark and recapture. So you'll mark some individuals and then you'll check back later and recapture some of these individuals and see how many of them are marked. And that gives you an idea of how many individuals you have in that particular area. How many are marked versus how many are not marked. You can do some math and figure out how many individuals you have. So with this, would you do this with mushrooms? Absolutely not. That doesn't make any sense. Would you do this with big predators like bald eagles? Yes, you could do it with them. There are lots of different species that you could use this with, but you wouldn't use this with stationary individuals. So what's so important about this is you have to figure out the right way to measure population size depending on the species that you're looking at. Wouldn't that make sense? I want you to go to your OpenStax Biology 2nd edition free textbook. If you haven't used it this term for this course, it's still free, so just go look at it. Chapter 45, first section, this is the ecology chapter, and this will tell you why you would use these different population measuring techniques, why you would use one over another and why you wouldn't use one of these, okay? For this exercise, you have a big sheet that you're going to use with different colored blocks on it. You can use the wax pencil that you have used already in this term on that laminated sheet and just wipe it off with a paper towel. Works perfectly fine. They say, I think, use a dry erase marker. You don't have to do that. Use the wax pencil that you already have. It'll come right off. And you're going to need to figure out when you answer the questions why you would use one counting technique over another one. And that's why I said, look at that chapter 45, section 1, and you'll have great examples there and you'll have all of this explained. Okay? So that's what you got going on this week. If you have any unfinished work, that is due by the last day of this course, which is Sunday, October 23rd, by 11:59 p.m. If you hit it at midnight, you cannot turn anything else in. So you got to turn in anything that you're going to turn in by that day and by that time. You can't send it to me to put into the course. I cannot do it. All I can do is grade. So make sure you have everything in by the last day of the course. I'll be inputting grades very shortly after that, either Monday or Tuesday of that week, next week. So you can be looking for those. One other thing is, if you would please fill out your course evaluation. That would be awesome because that helps the people who supervise me, my team leader and my dean, to evaluate my performance and how the course is working out for everybody. You also talk about the resources like your OpenStax textbook and the labs that are written for you, that sort of thing, plus me. Okay, I would appreciate it very much if you would fill those out for me. And as always, if you need anything, there I am, m.sigmon@snhu.edu. Please let me know if you need anything this week. This is a really important week to get through. So let me know if you need anything. Have a great week.